everyone, and welcome to AP Help 101. So this week I'm going to start talking about heredity, and heredity is a really big unit, so I'm going to actually break it up into two different videos. So just a really brief introduction about heredity. Gregor Mendel, he's the father of modern genetics, and he actually had his own pea plant experiment where he discovered alleles and genes and so on and so forth. And just another definition, particulate inheritance is when inherited characteristics are carried by discrete units called genes. And that's another thing Gregor Mendel also discovered. So there are a couple of laws to remember. The first one is the law of dominance, and this was Mendel's law. It's also known as Mendel's first law, actually, and basically what it means is when two organisms are crossed, both of them being homozygous, then the offspring will be a hybrid, having two different alleles. So what this is if you have one organism that has two dominant alleles and the other one having two recessive alleles, and you cross them together, their offspring all will have one dominant and one recessive allele. But in that case, the dominant trait, that trait will be exhibited because each one of them has one dominant and one recessive. A recessive trait is a trait that is hidden unless there are two recessive alleles. We also have the law of segregation, and what that means is that during the formation of gametes, the traits carried by each parent separate. So we have the parent cell, which has a dominant allele and a recessive allele. And when these sex cells are created, one sex cell will have the dominant trait and the other one will have the recessive trait. A backcross or test cross, which is a way to determine the genotype of an individual plant or animal showing only the dominant trait. That's just a definition that you need to know. And another law, there's a lot of laws, the law of independent assortment. And what that means is it's applied when a cross is carried out between two individuals which are hybrid for two or more traits that are not on the same chromosome. This is also known as a dihybrid cross. So basically, what happens is alleles of a gene for one trait will segregate, or they'll separate independently from other genes. So for example, height and seed color of a plant are not related in any way with each other. So they will separate independently from other genes. If the genes are linked, or if they're on the same chromosome, then they will not separate independently. They will separate together. So just some other inheritance topics that are important to remember. We have incomplete dominance, which is also known as blending. So a good example of this would be if you have a red flower crossed with a white flower and they produce pink flowers. Codominance is another one. That's when both traits are shown. So in humans we have MN blood groups, which are different from our ABO blood groups actually. And basically there are three possibilities. You have MM, NN, or a combination of M and N. And M and N is not really an in-between state. It means that both molecules of M and N are on the cells. And multiple alleles is when there are more than two allelic forms of a gene. And a good example of that would be your ABO blood groups. So you either have A, B, AB, or O. And A and B are codominant. So again, when you have if you have AB blood, for example, you will show the molecules from A and B on your red blood cells. And finally, we have some gene interactions. Pleiotropy is the ability of one single gene to affect an organism in several or many ways. So a good example of this would be cystic fibrosis, which is an autosomal recessive disease. It has many different side effects, not just affecting one part of the body, but multiple parts. Cystic fibrosis usually leads to mucus buildup in lungs, the digestive tract, and in all other organs in the body. It also leads to poor absorption of nutrients and perhaps bronchitis as well. So as you can see, this one gene is affecting this organism in multiple ways in multiple organ systems. An epistasis is when two separate genes control one trait, but one gene masks the expression of the other gene. So this one's a little more complex and it's a little more difficult to explain here, but I have a really good example of epistasis. So in my example, we have the agouti coat color in mice, and this is a dihybrid cross. So if we had a simple dominance, then it'd be a 9 to 3 to 3 to 1 ratio. But we know here in this case that both gene A and gene B are required to produce the agouti color in the mice. Okay, well now that complicates things a little bit, because now we'll only see three different color combinations. We have the agouti, we have the black, and we have albino. But here in this case, even in the absence of gene B, even if A is present, the coat is still albino. We still don't get that agouti color. You need to have both gene A and gene B dominant to be able to see that agouti color. So in this case, we say that gene B is epistatic to gene A because it's masking the expression of gene A. Little complex, but epistasis is really just when we have two genes and it does control one trait, but one gene masks the expression of the other gene. So I hope you understand this and get a hold of Heredity. There's a lot more in Heredity, and so I'll be doing another PowerPoint later to finish off Heredity Part 2. So if you like this, if you have any suggestions, place them in the comments below and hit that subscribe button, and I'll see you guys next week.